We're here today to listen to the thesis defense of student Brendon, who's been here, I don't know how many years, 5 years now? Is it 5 years? Four and a half?
Sorry, I was muted. Yeah, 5 years.
That's 5 years. So he's fortunately, I guess, going off to work at NASA, I think. Is that correct?
Mm-hmm.
So we're looking forward to successful. I think you're going to be with Lewis's group, right? Is that correct? Or...
Yeah, most likely in that same branch.
So that's exciting news. So his work today so his thesis has taken a long circuitous path from starting with the immune system and trying to figure out how to handle this very complicated and poorly understood process by which our body decides if it's going to kill itself or not. So hopefully we've made some progress understanding it, although perhaps we've just touched on it. Perhaps the progress will continue. In any case, in that quest, I think he's made lots of interesting discoveries, trying to use data to understand and manipulate the complex models, nonlinear systems. So we hope to hear all about that today. So with that, I'll turn it over to you, Brendon.
Okay, yeah, thank you for that introduction, Dr. Pete. So as he was saying, today I'm going to be talking about how we can generate predictive models from data, specifically how we can use that to analyze and identify components of the immune system. So I think let's see, there we go. So I think an important question is, well, why do we actually care about the immune system? And the answer to that is that it has some very important roles in the body. For instance, the immune system, a healthy immune system at least, is required to eliminate harmful, what we call non-self. Now, non-self can be something like a virus that we see in figure 1 or a bacteria. And without the immune system to sort of handle these threats, you would of course be very susceptible to diseases. On top of that, the body also has to be able to protect self, right? It can't just attack everything in the body. It has to be able to identify between what should be protected and what should be tolerated versus what should be eliminated. For instance, we have a red blood cell here, which obviously if your body attacked those would cause a lot of issues. So when we have a malfunctioning immune system, we have issues such as the body can protect harmful non-self. It can sort of ignore viruses or bacteria, and that leads to susceptibility, to where you'd be susceptible to these diseases. Another issue is if your body tries to eliminate itself, then that leads to autoimmunity, which can cause ailments such as diabetes or rheumatoid arthritis. This identification problem is actually somewhat difficult, especially when you consider cases such as cancer, where cancer cells are technically sort of yourself because they're made out of your own cells, but really they're non-self. They're harmful to the body and they should be eliminated, and they can be eliminated by the immune system, but they aren't always effectively or as effectively eliminated as they should be. So again, that leads us all to this larger question of how does the immune system actually identify self versus non-self? So if we were to try and predict the immune response, then we need to kind of understand how this self versus non-self determination can occur. And one of the issues with determining what the response will be is that there are 25 billion white blood cells alone in your body, and there's not really any central control system that tells them what to do. They sort of collectively have to identify whether something is a threat, whether it's non-self, or whether it's self, and then whether it should protect or ignore the self, and then eliminate the non-self. And the way this works is you have classes, or you have immune cells that have different roles, right? So you have regulatory cells, and they sort of dampen the immune response. They say when they identify something as self, they essentially tell the immune system to ignore it, to not attack it. So you have that class of cells, but then you also have inflammatory or effector cells. And those, the job of those cells is to eliminate non-self. But of course, if you have too many of those, or if they're identifying the wrong things as self, that's when you can have things like autoimmunity. And I guess the basic idea of this slide is that we need to understand the role of the cells to be able to determine how they're going to interact with the things they identify, whether they're going to identify as self or as non-self. Now, how do we actually determine what the role of these cells are? Well, we have to use one of the ways that these immune system cells interact are generally through these cell surface receptors, right? They can react to molecules in the body that were released from other immune cells, or they can directly interact using these surface receptors to other cells. So if we can measure what surface receptors the cells have and how many they have, that gives us an idea of what the role in the body is. For example, CD8 is a cellular molecule that we can measure, and that's usually associated with a type of cell called cytotoxic T cells. And what these T cells do is they actually eliminate cells in the body that have been infected with a virus to help slow the spread of that virus. So if we can measure a cell and we see that it has CD8, then we can assume or we can have a better understanding of what that cell's role is in the body. Now, how do we actually measure these cells? We use a process called flow cytometry. Now, if you have a solution of cells, then you can mix a set of antibodies in with these cells. On the right in figure 4 here, we have a diagram of that. So this right here is an antibody, the green and orange shape. And on one end, the antibody is specific for, in this case, a surface receptor. Let's just say this is, in fact, CD8. So that means that when you add these antibodies to the mix of cells, they're going to be attracted to and connect to these surface receptors, these CD8 surface receptors. And then what happens is the flow cytometry device puts the cells into a single column. And as they go down the column, you have lasers that excite the back part of this molecule, of the antibody, which is a fluorescent molecule. So when the laser hits this molecule, it fluoresces and releases, let's just say, orange light in this case. And the flow cytometer has photodiodes that can record the fluorescence that's returned by the antibody after it's been hit by the laser. So if we see a lot of orange light, that means that that cell has a lot of CD8. And then again, because we know what the purpose of CD8 is, we can determine what type of cell is and what the role of the cell is. So how do we actually use this flow cytometry data? Well, one of the traditional ways is you label these data sets as either being positive or negative, or you label the groups of these cells as being either positive or negative for the cell receptors. So here we have a diagram of CD66 and CD11B. And on this slide, you actually see millions of cells. Each of these black dots is an individual cell that was measured, or that had these CD11B and CD66 receptors measured. And now what you can do is, for instance, in the red box, you have cells that are high for CD66 and have high levels of CD11B. So those would be CD66 positive and CD11B positive cells. In the green box, on the other hand, you have cells that are low for CD66 and low for CD11B. So we would call those CD66 negative and CD11B positive. And there are many different methods of actually grouping these cells. The one I have shown here isn't necessarily the only way or the best way, but usually these are done by hand by looking at these 2D plots and finding groups of cells. The problems with sort of analyzing flow cytometry data in this way is, how do we know which cell characteristics are most important? You can measure dozens of these different surface receptors or molecules on the cells. And it's not entirely clear, just looking at the plots, which ones are most important and which ones should be studied further. So that's one of the problems we're going to look at today. The next one is what cell populations are most relevant to a disease. So after we've grouped these cells by by their measured cell receptors, we're assuming that, for instance, all the cells in the red box have a similar role because they all have similar characteristics. So when we group these cells into these similar roles and measure them, well, which populations do we need to look at? Which ones are most relevant to a given disease? To again, either be studied more or to determine why something such as rheumatoid arthritis is occurring in the patient. And then finally, we're going to look at how these populations influence the effectiveness of immunotherapies. There are many different types of immunotherapies, and they all act to sort of correct a malfunctioning immune system, or to, for instance, with vaccines, to train the immune system in a new way, so that we don't have to, say, get sick to become immune to a disease. So the population of immune cells in your body is going to affect how effective these treatments actually are. So we're going to look at how we can use information on these populations to generate better immunotherapies. And to do so, we're going to develop a few different data-driven methods that can solve these problems. But first, I think it's important to talk about what actually is a data-driven method. Well, data-driven methods, as the name would imply, take in data, and then they generate functions that need to model some process, ideally the process that generated the data. These usually consist of three components, and that's how we're going to define them. The first component is we need data, of course, and we have an example of some simple input-output data here. And we assume that this data comes from a data generating mechanism, which is just the physical process that generated the data. So if we're looking at flow cytometry data, that would actually be the individual that the cells came from. They sort of generate these immune system cells, and you can measure the cells from that person. So they themselves would sort of be the data generating mechanism. Next, you need a set of candidate functions, which I like to call a bag. So we have this bag of functions here. And some of the functions inside do a good job of capturing the trends in the data. For instance, we have this g of x function, which generates this red function. And we see that that fits the data very well. However, we also have this f of x function in here, this blue function, that fits the data very poorly. Now, this isn't necessarily a problem. We want to have a wide variety of candidate functions. But to use this effectively, we also need an algorithm that chooses the best candidate. We need something that can look at all the functions in our bag and decide that, yes, in fact, this g of x function, this red function, fits the data best, and that's the one we should use to model our data. Okay, so each of these are data-driven methods that we'll be talking about, or that I'll be talking about today in this talk. And we're going to start off with performing uncertainty quantification with positive polynomials. Now, why are we interested in uncertainty quantification? Well, let's just say we're given points, delta, and we're given m of them. In the case of flow cytometry, you're given oftentimes hundreds of thousands, potentially even millions of points. And we assume that these points are drawn from some data generating mechanism. What we want to do is we want to model the data generating mechanism with a probability density function, right? So if we look at these examples, we have figure 6 here and figure 7. These don't necessarily, to the naked eye, you can kind of see some differences. For instance, you see that there's some cells here that seem to be missing in figure 6. However, you can't exactly tell, well, where are most of the cells? There's just too many cells to visually look at. But if you model the probability density of these cells, then you can see quite easily what the differences between these two populations are. In figure 6, you have sort of this smaller, higher density area of cells that are high for CD66 and low for CD11b. And over here, you have a population that's low for both. And on this other patient, you see that they have an area that has higher CD11b and higher CD66 than if you just looked at the data alone. So this probability density function tells you a lot about the data itself. So what actually are probability density functions? Well, a function f is a probability density function, or a PDF, if the integral over its domain delta is equal to 1. And this is important because the domain is all the places where a random point could be drawn from. So of course, the probability of drawing a point from your domain must be 100%. So this constraint here enforces that property. In addition, the PDF must be greater than or equal to 0 on the domain as well. And that enforces the constraint that you can't have a negative probability, because that doesn't make sense. All probabilities have to be positive, and they must be less than or equal to 100% when you're looking at PDFs. And that is because of this property, that the probability of a point over a set is exactly the integral of the PDF over that set X. So the probability of X is the integral of this function over the set X. Now, as an example, if we look at a simple PDF of a Gaussian, which we see here in figure 8, if we can define the probabilities of a point being drawn between, let's say, 0 and 1 standard deviation, away from the mean, which in this case is 0. And we see that that is exactly 34.1%. So that's sort of where the PDF becomes useful, in that you can characterize where future random points are likely to be drawn from. Okay, so what do we do to improve this uncertainty quantification process? Well, we noticed that two common PDFs are the exponential PDF, which has a linear polynomial argument to an exponential. So we have a linear polynomial, which means that the polynomial has a degree equal to 1. And a normal or a Gaussian PDF. We see that's essentially the same form, except instead of having a degree 1 polynomial, you have a degree 2 polynomial. In the we have an exponential of the degree 2 polynomial. So the question is, well, what about a degree 3 or a degree 4 polynomial? If we sort of increase the degree of these polynomials, can we model significantly more complex PDFs than just an exponential or normal PDF can? It turns out that we can. Here we have a degree 2 slice PDF in this first column. We look just at this first column on the left. Here, the PDF of a Gaussian says that the highest probability of points should be in this center area that I'm sort of highlighting. But if we look at the actual data, which is down below, there's actually no points in this central area. So the Gaussian PDF does a very poor job of modeling sort of the density of these points. However, if we bump it up to a degree 4 slice PDF, so instead of having a quadratic polynomial, we use a quartic polynomial, you see that the highest density areas are up in these two corners of the circle, which is exactly where most of the data is on the plot. And if we go all the way to a degree 16 slice PDF, we see that we can fit the data extremely tightly. These sets you see that contain 25%, 50%, 75%, and 100% of the data, all very tightly conform to the data. So you can quantify where future points are most likely to fall without actually including a bunch of wasted space that is actually where points are actually unlikely to fall. So the properties of the slice distributions are that they are dense in all bounded PDFs. We can select these using convex optimization. And what that means is that bag, that candidate of candidate functions, if we fill that bag with slice distributions, we have an algorithm that can always find the one that is most optimal within that bag to model our data. And then finally, the level sets of these slice distributions are also semi-algebraic sets, which mean they can be applied to additional optimization programs such as sum of squares programming. And then finally, we developed several different optimization methods to select these slice distributions. The last one is always guaranteed to find the best slice exponential from within the bag, and the other two are suboptimal selection criteria, but they can be performed very quickly and still have a relatively good fit to the data. Now, to just show that we can characterize higher dimensional distributions of points, in figure 10, we show some original data that was sampled from just a synthetic data generating mechanism. And then we simulated that data using a degree 4 slice distribution. And you see that the data is captured fairly well. You see that the curves in the data are captured in the simulated data points, and that the overall shapes appear to be very similar between the original data and the simulated data. So even in these higher dimensional spaces that are more difficult to sort of visualize, we can still capture these distributions rather well. Now, how are we going to use this to analyze the immune system? Well, we have an unlabeled rheumatoid arthritis data set where we have flow cytometry data with 41 different cell attributes, so 41 different surface receptors or other molecular markers that we can measure on the cell. And then we have samples from 5 patients that do not have rheumatoid arthritis for a total of a little over 1 million points, or 1 million different measured cells. And then we have samples from 9 patients that do have rheumatoid arthritis for a total of about 3 million different cells. And again, the question that we're trying to answer is, what cellular characteristics are going to be most correlated to healthy patients or those with rheumatoid arthritis? And the problems we're going to try and solve is that sorting these cell populations can be very time consuming. And we can only sort them by 2 characteristics at a time, because at any given time, you can only look at a plot that has 2 dimensions. So the question is, can we try and automate this flow cytometry data to make it easier to find interesting populations of cells much more quickly? So how are we going to do this? We're going to identify, or again, we wanted to identify the cell markers that differ most between the patients that are healthy and the patients that have rheumatoid arthritis. So what we're going to do is we're going to model the distribution of all the healthy cells of all of the patients. We're going to call that model FH. We're going to model the probability density function of all of the cells from patients with rheumatoid arthritis. And we're going to call that model FR. Then we're going to use a feature selection technique, and I'll discuss what feature selection is on the next slide, to find the cell characteristics where they differ the most. And then to show that the PDF correctly captured the characteristics of these cells, we're going to use the models to differentiate between the healthy patients and those that have rheumatoid arthritis. Now, a question though is, how do we determine how similar 2 models are, right? So we have 2 of these models. We have FH and FR to model the healthy cells and the cells that have from a patient with rheumatoid arthritis. And the question is, how do we know if these are similar or not? And to do that, we'll use the Hellinger distance. And the main takeaway of the Hellinger distance is that if both of the PDFs are exactly the same, then the distance is 0. And if the distribution, or if the PDFs are non-zero only, or if let's say FR is non-zero, then the healthy PDF must also equal 0. So essentially, neither populations are non-zero at the same point. If that's the case, then the distance is equal to 1. So our distance is bounded between 0 and 1. And since we want to find where they are most different, we're looking to try and find the cell characteristics that lead us to a Hellinger distance that's as close to 1 as possible. Okay, so how do we perform feature selection? Well, instead of having a bag of, say, candidate functions, in this case, we have a bag of features of cellular characteristics. We want to find which ones generate models that have the largest Hellinger distance. Now, the way that the sequential forward selection algorithm that we use works is we start with the empty set, and then we test each of the attributes individually. So let's say we use CD32 first, and that gives us a Hellinger distance of 0.1. And then we test CD66, and that gives us a Hellinger distance of 0.5. And then we test CD8, and we get a Hellinger distance of 0.7. Then we would add CD8 to our set. So now S would equal CD8. And then we would start this process all over again, except instead of testing each feature individually, we would instead add each of these features that are remaining to CD8 and see which ones lead to the largest Hellinger distance. And we repeat that n times until we have the number of n attributes that we want to select. In this case, we selected 3. So again, we did this on the healthy immune, or we made these models for the healthy immune data and the immune data from patients with rheumatoid arthritis. And we found that CD66, CD11b, and a DNA intercalator ended up being the most important cellular characteristics to look at. We're going to show the PDFs of these 2 distributions, and we see that the overall distributions of all the cells from the healthy individuals and those from those who have rheumatoid arthritis vary quite significantly when you look at these PDFs. This entire sort of subpopulation here was non-existent in the healthy, or in patients that were healthy when compared to those with rheumatoid arthritis. Again, to show that these models correctly differentiate between patients that are healthy and those that have rheumatoid arthritis, we tested each of the patients individually to see if their data was more likely to have been generated by the healthy model or the rheumatoid arthritis model. And in all but one case, they were correctly identified. So in one case, a healthy patient was actually more likely, their flow cytometry data was more likely to have been generated by this rheumatoid arthritis immune model. But overall, it seems to have captured the differences between the cell populations very well. So in conclusion, we can use slice distributions to model these data generating mechanisms. We can use them to simulate new samples. We're able to find sets that tightly contain delta, and that can be used for robust modeling using, as I said earlier, sum of squares programming. And finally, we applied that to identify the cellular characteristics that differed most between healthy patients and those with rheumatoid arthritis. Now, important properties of the distributions are that we make no assumptions on the key features of the DGM. Since slice distributions are dense and all bounded PDFs, we can model extremely complex data generating mechanisms. And we don't have to assume things such as when you use a Gaussian that the data is normally distributed. In addition, if we increase the degree, in general, that allows us to increase the complexity of the DGM that we are actually measuring. So usually, or that we're trying to model, sorry. So increasing the degree allows us to model increasingly complex data generating mechanisms. Okay. Next, we're going to talk about a second data-driven method that was formulated, which is how can we generate better machine learning algorithms using positive kernels and positive matrices. So first, I just want to go back and recall what the components of the data-driven methods are. Well, first you need, again, data. However, since we're trying to formulate a better machine learning algorithm, we're assuming we're just given the data. We have no choice over how good or how bad the data is. So instead, we're going to look at, well, how do we create better sets of candidate functions? And then how can we generate good algorithms so that we can always choose the best candidate from our bag of candidates? So the main idea behind this is, well, let's use the data itself to generate the candidate functions that we're then going to search over. And then we're going to use convex optimization to both select the candidate functions and also select the best candidate function from the bag. So we're going to basically use the data itself to tell us what our bag of candidate functions should look like. And we're going to do this by building off of kernel-based machine learning algorithms. So we assume again that we're given data. We're given m data points xi, and we're going to, and in these cases, kernel methods generate candidates or sets of candidate functions f, which is defined here. And really what each of these functions are is a linear combination of the kernel that's selected, analyzed at each data point, right? So again, since we can't choose better data to change the set of candidate functions, really all we can do is look at this kernel function k. And the kernel function k can't be any function. It has to satisfy this constraint. So it's not exactly a trivial problem to just come up with a good kernel function. So one of the methods that was developed to help people select better kernel functions is called kernel learning methods. And what these do is they search for an optimal kernel function k from our set of kernel functions. What was not really addressed before we started researching into this, though, was, well, what makes a set of kernels optimal, right? Just because you can make a set of kernels and search through them doesn't necessarily mean that that is a good set of kernels to make. So we want to look into what are the properties of an optimal set of kernel functions, and then can we actually propose a set of kernels k that is optimal, that sort of follows this criteria we've given. So the desired properties of a set of kernels k is first that it is universal. And what that means is that the set of models is dense for any of the kernels in k. And I'll describe what density means in the next slide. Tractable is the next condition. And that just means that the set of kernels has a linear parameterization, which means that the problem is convex, which means that we can always select the best kernel from our set of kernels. So that's part of that third criteria for a data-driven algorithm of you want to be able to search your set of candidate functions and select the best one. And finally, we have this density property, which implies that the set of kernels k is dense in the set of all kernel functions. And really what this just means is that you could give me any kernel function and I could search through the sort of set of candidate kernel functions in k, and I can find one that is arbitrarily close to that kernel function. Now, there are some classes of kernels that have some of these properties. For instance, there are universal kernels or sets of kernels, but that have poor performance because they're not tractable or they are not dense. And then there are also trivial classes of tractable kernels that are easy to search over, but don't necessarily perform well. And then there's also dense kernels, but they may be impossible to search over. So we're looking for a class of kernels that has all three properties. And as of yet, well, until the one that we're going to propose, we have not found a class of kernels that can do these three things. Now, why is the universality property important? Well, a set of kernels are universal if for any kernel function from our set of kernels, so we can select any of them, the set of candidate functions f is dense in L2. And what that means in practice is that you can essentially give the algorithm as complex of a set of data as you want, and it'll still be able to accurately model the data. So here on the left in figure 15, we have a non-universal kernel that is trying to classify these data points. And what classification is it wants to label these red X's as being red, which is you see in this highlighted red area. And it wants to highlight the blue points or the blue circles as blue. So in both these cases, on the left, we have the non-universal kernel, and on the right, we have the universal. We see that both kernels do a good job of modeling this data. The blue points are in the blue highlighted area, and the red points are in the red highlighted area. But what if we make it more complex? Well, if we repeat the same data and just shift it over one unit, what we see is that the non-universal kernel can still model the data fairly well, and so can the universal kernel. But if we do it a third time, the non-universal kernel fails and classifies some of these blue circles as actually being in the red area. So the non-universal kernel has failed while the universal kernel has succeeded. And of course, we can do it again and the universal kernel will continue to fit the data perfectly. And we could continue this pattern an infinite number of times, and a universal kernel is still able to accurately predict or accurately model this relationship. So that's kind of the importance of this universality property, because that means that any of the kernels K within our set can do this. Next, why is the density property important? Well, again, you can give me any kernel, let's call it K star, and then a kernel K exists within our set of kernels that is arbitrarily close to the kernel that you gave to me. Now, what this implies is that if an optimal kernel exists, let's say K star is in fact the optimal kernel, well, then we have a kernel within our set that is arbitrarily close to that optimal kernel. And this fact is independent of the data points X and our labels Y, which means, again, this is independent of the data because we can't choose how good or how bad our data is. So it's important that these properties are independent of that data. So the kernel function or the class of kernel functions that we propose is called the class of tessellated kernel functions. It looks like this. And the main thing that we can choose to change how the kernel function behaves is we change this matrix P. This term on the left and the right is all related to manipulating the data. So we have ZDZX being a monomial basis, and this indicator function, which is 1 if your point is in the positive orthant. Now, of course, the question is, well, is every TK kernel a positive kernel? Again, if the kernel isn't positive, then you can't actually use it as a kernel function. And then next is the question is, well, is this TK kernel optimal? Does it meet those 3 criteria that we've just discussed? Well, we've shown that for any positive semi-definite P, the TK kernels are positive, tractable, dense, and universal. In addition, we proposed efficient methods for selecting the best tessellated kernel function. Now here you have sort of a visual example of what the learned function using a TK function or using a tessellated kernel looks like. And in this case, it is a piecewise linear function whose domain is broken up by each of the data points. So you see that at this data point, the slope of this line can change. And that depends on the tessellated kernel function that is selected. Can change those properties. Now we did a numerical analysis to compare to other machine learning algorithms on a set of 12 different data sets. Here we're showing 3 of the regression problems. And we see that in this case, using the tessellated kernel learning algorithm that we developed outperformed all the other methods except for on the CCPP data set where we performed second. Meanwhile, on classification problems, which is when your output is binary, like that diagram of the X's, the red X's and the blue O's that I showed earlier, TKL did best on one of the data sets and did second best on 2 of the other data sets that we're showing here. And then on all 12 of the data sets that we did analysis on, TKL performed above average on all but 1 of those 12 data sets, which again indicates that while no machine learning algorithm is always going to perform best on any set of data, what TKL does is it's most consistent when compared to these other machine learning algorithms. Now we applied these machine learning techniques to a labeled rheumatoid arthritis data set, which had 42 labeled immune features that were labeled by the traditional cell sorting. And then we also used the location the cells were collected from, and also a treatment that were given to the mice as an input to the machine learning algorithms as well. And then finally, we also had a rheumatoid arthritis score, which is a way of measuring the severity of the disease. Now, how was this data generated? Well, first, rheumatoid arthritis was induced in the mice by an injection of bovine collagen II autoantigen with 2 adjuvants, CFA and IFA on day 0 and day 21. And then a treatment was applied of either the actual immunotherapy treatment or a control treatment on day 35 and 42. And then finally on day 70, the cells were measured using flow cytometry. And because some of the mice were given control treatments and some were given an actual immunotherapy, the severity at the end of these tests differed because the immunotherapy actually performed rather well. And the question that we're asking is, how do we identify the important populations using these machine learning techniques that are most relevant to, say, the severity of the disease? So again, the main idea here is to, again, use feature selection to identify the immune cell populations that first can best predict other immune system populations. Now, why is this first sort of bullet important? Well, there are hundreds of potential immune system populations that may actually be important or relevant to the disease. Or sorry, there are hundreds of immune system populations, but only a few are likely going to be relevant to the disease. So we want to find the immune system cells that can best sort of distill the information of all of these immune system populations and are sort of are most relevant to controlling the immune system as a whole. In addition, we want to track the disease progression. And of course, this is useful because it gives us sort of a metric of how bad the disease is, and by seeing which cells cause the disease to be worse, kind of tells us which cells drive the disease. Now, again, feature selection is really all about having a bag or a candidate of features. In this case, it is the cell populations. And then generating models and figuring out which of the immune features, in this case, the second immune feature we're saying produces a good model, whereas the first immune feature made a poor model. We want to find all of these features that generate good models. But again, the question is, well, what makes a model good and what makes it bad? Well, we use a feature selection metric that does three things. The first is we want to be able to predict the disease severity well using a machine learning algorithm FS, or a machine learning learned function FS. We want to be able to predict the discarded features FD, or using the trained function FD. And then finally, again, we want to encourage a minimal representation of immune system cells that are most relevant to the disease. So we also want to make sure that B only contains a few features. So the actual feature selection metric is broken up into these three parts. If we let R of F and X and Y be the root mean square error of the predictor F using the inputs X and the outputs Y, we want to find the features that minimizes sort of metric 1, which again is how well it can predict the disease severity. Metric 2, which is how well it can discard features. And then metric 3, which is really just a way of ensuring that the number of features B is less than or equal to 10. And we are going to analyze this metric on what we call a validation set. And what the validation set is we withhold that data and we train these functions FS and FD using the remaining data. And then we test these functions on that validation set to get a more accurate estimate of how well these functions model the data on data it hasn't seen before, right? We want to know if it is generalized to the overall patterns of the data. Again, we perform that sequential forward selection technique that we used in the previous section. So we model the immune features separately, and then add the best one to the set S, and then continue iterating this n times, which in our case is 10, until these 10 populations have been selected. Now, to ensure that the immune cell populations that we have selected are in fact the most relevant to the disease, we actually applied 5 different machine learning algorithms to this problem, not just our Tessellated kernel learning algorithm. And then we wanted to see which of or where all of these algorithms sort of agreed the important cells were. So we ended up with a few different plots that looked like this, where here we have cell populations. that are highlighted if at least a majority of the algorithms, or sorry, are bolded, if at least a majority of the algorithms selected that cell population as being important. And it selected quite a few cells as being important, but we want to highlight just a few of them. Perhaps the two most important were the memory effector cell, was a memory effector cell population. And what memory effector cells do is they are normal effector cells, so they produce more of an inflammatory response, but they have memory. They can remember previous threats that they've encountered. So, and because they've encountered them previously, they can react quicker. So it makes sense that these memory effector cell populations would be very important to the disease because they're going to be able to act the quickest and generate the response. We also have a population of cytotoxic effector cells that was selected very often by all of the algorithms. And this again also makes sense because these are the effector cells that are actually essentially causing the damage. They're actually attacking the cells or the body's own cells. Something else that was important was that the location the cells were collected from was not actually selected as being important for determining the disease severity. This is interesting because it means you can measure cells from essentially anywhere on the mouse, and we could still use these algorithms to predict how severe the disease is. So sort of the immune populations seem to be uniformly distributed. Now, in terms of the algorithm, we showed that the tessellated kernel function that we developed, that class, has the universality property, the tractability property, and the density property. And we showed that we could efficiently find the best kernel from within this class. Okay, so next we're going to talk about another sort of machine learning based idea, which is function fitting with positive polynomials. Now, why would we be interested in modeling with positive models? Well, sometimes you know that your data is in fact positive. For instance, if we assume that we have point estimates of a converse Lyapunov function, well, as can be seen in figure 21, well, converse Lyapunov functions are globally positive. So the model, the set of candidate functions that we want to use to model this data should also be positive functions. Because again, we want a model that can use this Lyapunov function to estimate the region of attraction of the system. So again, the problem is that we can't use a generic ML algorithm because we need to add this additional constraint of positivity. So we need to select a special set of functions to work as our candidate functions that are globally positive. And then finally, we're also going to talk about how we can find these point estimates that I talked about of the converse Lyapunov function when we don't actually know the system dynamics. So for this problem, we're going to assume that we have trajectories of an unknown system. So here on figure 22, we see the vector field of the van der Waals oscillator. And then we also see two trajectories starting from here and here that end up going into the equilibrium point of this VDP oscillator. Now we're going to assume that we don't know this vector field. We don't know how the system dynamics actually work, but they were only given a few of these trajectory measurements. And then we want to know, well, is that point going to also sort of go to the equilibrium point? Is that also in the region of attraction? Or what about this point over here? We want to calculate a model of the region of attraction using only these sort of trajectory information. Now, the problem is that we can't search for a Lyapunov function without knowing the vector field. However, we can solve this by choosing to model a Lyapunov function that is defined by trajectory information. So a form of converse Lyapunov function, v star of x, is actually equal to the integral of the trajectory, or of g of x of t, which is the trajectory throughout time, squared. So if we have measurements of the state of a system throughout time, we can actually estimate this integral for the starting condition of our trajectory. Now, this converse Lyapunov function satisfies the constraints of the Lyapunov function that v star of x, or so that it's globally positive, and that the time derivative is 0 within the region of attraction, or is less than 0 within the region of attraction. And if we have a model of the Lyapunov function, then we can estimate the region of attraction as a level set of the converse Lyapunov function itself. And if we have the exact converse Lyapunov function, then the region of attraction is exactly this level set as c goes to infinity. In our case, we won't have the exact level set, so we do have to choose a level set value to estimate this region of attraction. So now we have the data that we're trying to fit, which is these converse Lyapunov function evaluated at the initial conditions. And we still need to find a set of functions that is globally positive to make up our bag of candidate functions we're going to select from. Now, again, the model requirements are that they must all be globally positive, and they also must be convex, because we want some method to choose the best candidate function from within that bag. And the solution to this problem is we're going to search over the set of degree-bounded sum of squares polynomials. Now, again, if we let z dx be the vector of monomials of degree d or less, and then we can parameterize any SOS polynomial as the following set of candidate functions, where the only requirement is that p is positive semi-definite. Now, all of these functions, by definition, are globally positive, since p is positive semi-definite, and the set of models of these candidate models is in fact convex as well. So to overview the proposed method, we're going to assume that we have no information on the actual system dynamics, but we do have some trajectory information. So we can estimate this integral at the initial conditions of our trajectories. Then what we're going to do is we're going to fit our sum of squares polynomial to minimize a norm between the value of the SOS polynomial model and our converse Lyapunov function values at the initial conditions, which really is just saying we're trying to minimize the error between the model we're selecting and the data that we have. And then finally, we can estimate the region of attraction as a level set of our sum of squares polynomial that we call RC with an appropriate choice of C. And usually what we choose for C is just the largest value of our estimated converse Lyapunov function points, just to ensure that our region of attraction contains all of the points that we know to be within the region of attraction because their trajectories ended at the equilibrium point. As an example of a learned Lyapunov function, here we have one for the Van der Pol oscillator that was in the that I showed close to the introduction of this method. That used only 50 trajectory measurements to generate this model. Now we're going to apply this to an immunotherapy cancer data set that we are going to simulate. So we have data that is simulated trajectories of the tumor cells using a cancer immunotherapy model that had 5 states. The first state is the volume of the tumor cells. Now what these tumor cells do is they secrete the cytokine TGF-beta. So they secrete this cytokine to increase the amount of TGF-beta. And then this TGF-beta inhibits the immune cells. So it inhibits these immune cells, E, and increases the proliferation of the tumor cells to increase the tumor growth. And then the effector cells trying to inhibit the tumor growth, but again, they are inhibited by the TGF-beta. And then to ensure that this immune system, or that this process is regulated, the effector cells also generate regulatory cells that in turn inhibit the effector cells, again, to regulate this response so that the effector cells don't go out of control and again start, for instance, attacking the body. And then finally, we have a vaccine and immunotherapy that can be used to inject mature effector cells into the body. So these are taken from the patient, they're matured outside the body and then re-injected into the patient. And this is done because it's assumed that these matured effector cells can again inhibit the tumor cells, but also aren't affected by the TGF beta because they're no longer proliferating. So essentially, what we can do is we can administer this treatment, and we're assuming that it's administered NV times at a dosage DV over 60 days. And the question is can we select a treatment that we know will lead to tumor elimination within those 60 days? And can we do that only using trajectory data, which in this case is data of how many tumor cells the individual had throughout time? Now we generate a region of attraction estimate using the model that we proposed, using 1,000 simulated trajectories. And again, we use that to generate an estimate of the region of attraction. Now what we want is a high true positive rate, because that means that if a selected treatment does in fact eliminate all of the tumor in 60 days, then we want to correctly predict that, that the treatment will work. And we want low false positive rates. And what a low false positive is, or sorry, what a false positive is when the model says that the treatment will work, but it actually doesn't. So we want low values of this term and high values of the true positive term. And we see that a 4th degree polynomial model of our region of attraction actually performed better than the 6th degree, which would imply that the 6th degree polynomial was overfitting to the data in some way. Now the question is, can we find an optimal treatment using this region of attraction estimate? So we take our Lyapunov function estimate, which is equal to ZDX times the optimal matrix P that we found, the P star. ZDX is our polynomial. And then gamma is the level set that we select. Then given the Lyapunov function estimate, or this Lyapunov function estimate, and the initial conditions of the tumor, the amount of TGF beta in the system, the amount of effector cells, and the amount of regulatory cells, we want to find the region of attraction, or we want to find the treatments such that this system is within the region of attraction, which means that the tumor cells will be completely eliminated within 60 days. So our gamma is exactly that set of points. So if you essentially just plug in NV and DV into this equation, and if it is in fact less than or equal to gamma, then you know that the treatment is predicted to work. But we don't want to just test random treatments. We want to actually see if we can use this to find an optimal treatment. Now, what would an optimal treatment be? Well, we want to avoid adverse effects. So what we tried to do was minimize the total treatment given, which is going to be the number of doses times the magnitude of the dose, which means that we want to solve this optimization problem, which is NV times DV, such that NV and DV fall within, again, this R gamma set. And because R gamma is a semi-algebraic set, this problem is known as a global polynomial optimization problem, which leads us to our final section, which is how can we actually solve global polynomial optimization problems? Now, a GPO is defined as an optimization problem where you try and find the minimal, or the point x, which minimizes a polynomial function f of x subject to constraints gi of x, where f and gi are all real valued polynomials. Now, aside from the application that I just gave, other applications of GPO problems are an economic dispatch, optimal power flow, and optimal decentralized control. Just to show how this method that we developed works, we're going to start with a simpler GPO problem. We're trying to find the minimum value of x2 such that these constraints are met. So what this problem looks like visually, we see in figure 25, we see that there are 3 disconnected sets that are feasible. And that means that these points meet these constraints. And we want to find the minimal value of x2 such that it falls within one of these feasible sets. Now, the problem with this is that since these sets are not connected, local solvers can often find a locally optimal point. For instance, they might stop here and might miss the globally optimal point, which is right there. So we did an analysis of local optimization methods. Some examples are sequential quadratic programming or interior point methods. And we found that they oftentimes can find locally optimal solutions. Since the problem is not convex, these local solvers are not able to find the globally optimal point. And we noted that these algorithms are going to be highly dependent on the starting conditions. So here we have that the globally optimal point is this red point right here. And we notice that if your starting condition is relatively close to the global optimum, then the local solvers generally find the right point. Whereas as you move farther away from the globally optimal point, the local solvers are unable to find the correct initial, or are unable to find the globally optimal point, which is this yellow area here. So our solution to this problem is, well, can we select better initial conditions using a convex optimization method? Now, a closely related problem to the GPO problem is called the greatest lower bound problem, or the GLB problem. And the way it's related is, instead of trying to find the point at which the function is minimized, you want to find what the minimal value of the function is at that point. So here we have lambda star. And what we're solving is we want to find the largest lambda such that f of x is greater than or equal to 0 on our set of feasible points. Visually in figure 27, if x star is this point right here, then lambda star is equal to this f of x star. So it's equal to a negative 1.4, whereas x star, that GPO, or that solving the GPO problem you are trying to find, that is actually about negative 0.4, right? So while lambda star equals f of x star, we don't know what x star is. The GLB problem doesn't tell us that. But we're going to use it to learn more information about the problem. Now, we're going to do that is we're going to bisect the feasible set. So here we've bisected that initial problem in half. And we've done that by adding one additional constraint to each of these two problems. So by adding this negative x times an x plus 30 greater than or equal to 0 constraint, we've constrained our feasible set to only be on the left side. Likewise, for this problem on the right, we've constrained it to be only on the right side. Now, how is this useful? Well, if we solve the GLB problem on both sides, what we find is, for instance, that x, or that lambda star on this side is about a negative 8, whereas lambda star on the right side is equal to about 2. Now, this tells us, since this is the greatest lower bound, that the optimal point can't possibly be on this right half, because the greatest lower bound is greater than the greatest lower bound on the left half. So we can discard this whole set, and we know that the optimal point can't belong there. Now, if we repeat this multiple times, continually discarding sets, for instance, we discard this set, and then next we'll discard this set, we get closer and closer, or the sets decrease in size. In fact, the longest edge of the hypercube decreases at a size, or at a rate of 2 to the negative q over n, where q is the number of iterations. And any point in the hypercube, the distance to the optimal point within the hypercube decreases by a factor of 1 over square root of 2 every n iterations. So essentially what this means is that the set that we're looking at is going to shrink around the optimal point. Now, unfortunately, the GLB problem cannot be solved exactly. There is a sum of squares tightening that's performed of the GLB problem, which essentially is solved by, we're trying to maximize the value of lambda such that f minus lambda is within m, where m is a sum of squares polynomial plus a sum of squares polynomial times each of your constraints. Now, since your constraints are polynomial, then m is, in fact, also a polynomial. So you can solve this problem. However, since you have to select a degree of your SOS polynomials, this problem is, again, not exact. It's conservative, which really means that lambda star is going to actually be less than or equal to f of x star. And here in figure 30, we can visually see this. Here we have a value of lambda star that is less than f of x star. But what we can also find is we can also find a locally optimal point. We'll call this XL. And then f of XL acts as an upper bound of the value of f of x star. So now we know that f of x star must fall within the set lambda star and f of XL. Now, how do we use this information? Well, we're going to use it in a similar way as the sort of optimal case when we assumed we could exactly solve the GLB problem. So here we've sort of updated the figure. We've bisected the set, and we've therefore found two different locally optimal points, one here and one there. And we've also found lower bounds on the value of f of x star on each side. Now, since the upper bound of f of x star is lower than the lower bound on this right side, we can also discard this set. So by using both lower bounds and locally optimal solvers, we're still able to discard sets and sort of decrease our hypercube to be closer and closer to the globally optimal point. Now, we applied this to that optimal immunotherapy problem from the previous section. Recall that we have this optimization problem where we're trying to minimize the total treatment given, which is NV times DV, subject to NV and DV being within our region of attraction estimate. We simulated these dynamics using an initial condition of, or using these initial conditions. And our GPO optimization problem gave us a dose of 3,472 of these effector cells to be given 17 times over the 60 days, which is approximately every 3.5 days. And if we look at the simulation, we see that it did, in fact, eliminate all the cancer cells within this 60-day mark. But we wanted to see how accurate this sort of method is on a lot of initial conditions. So here we simulated 10,000 different initial conditions, randomly sampled within these bounds that we see here. And we found that 85% of the selected treatments did in fact eliminate the tumor, and that 84% of the treatments were within 10% of the optimal treatment. And the way we calculated that was we decreased the dosage and increased the period of treatment by 10% to see if it still was able to eliminate the tumor within 60 days. And only 1% of the treatments were still able to eliminate the tumor in that case. Now, this is not a analysis of the GPO algorithm itself. Given the set R gamma that it was given, it did in fact find the optimal treatment. But because R gamma was, our region of attraction estimate was calculated using a data-driven method, this is more looking at errors in the region of attraction. So in conclusion, we developed a region of attraction algorithm that was able to find points that are in the region of attraction using just trajectories. So it required no information on the actual system dynamics. And we can do so efficiently, solving for thousands of trajectories in just seconds. And the method requires no particular structure of the ODEF. We also showed that the GPO algorithm can solve these global polynomial optimization problems, and that it is solvable in a finite number of iterations in polynomial time. And the tolerance of this solution is going to be dependent on the degree of the SOS tightening of the greatest lower bound problem. So in summary, we have proposed 4 different methods to characterize the uncertainty in measured data, to select optimal kernel functions for machine learning, to generate models with sum of squares polynomial functions, and finally to solve global polynomial optimization problems so that we can solve 3 of these immune system problems. The first being identifying the cellular characteristics that differ most between healthy immune systems and patients that had rheumatoid arthritis. The next was identifying important immune system cell populations that were relevant to rheumatoid arthritis. And then finally was determining the optimal post-immunotherapy treatments to eliminate cancer cells in a patient. Thank you for attending this talk.